occur and why the variations occur and how they get accumulated over the period of the time, right? Now, first of all, as I've discussed about that, hereditary means the transmission of the characteristics or the traits from their parents to their offspring. That's what we call as transmission. That's what we call as hereditary. And variation, as I've told you, is the difference in the characteristics which needed to be transmitted from one generation to the another generation. Now, what is the way of, just a moment. Okay, so I was discussing about the variation. How do these variation occur? What is the method of these variations? How do these variation occur? I told you that when organism reproduce, the offspring will definitely show some minor changes in, maybe in terms of body design. Maybe we have already discussed about the DNA inaccuracies, which are the basic event of reproduction. The basic event of reproduction is not the fusion between the two gametes. It is the basic event of reproduction, which we have already discussed about, is that it is copying of DNA. During that copying of DNA, although DNA tried to create an exact replica of itself, right? But as I've told you that DNA copying take place through biochemical reactions, and no biochemical reaction is free of error. So that's why we can say that during that time, whatever inaccuracies are caused during the copying of DNA, that gives rise to the variation in case of there is the asexual mode of reproduction. So in the asexual mode of reproduction, the variation is very less. But in terms of the sexual mode of reproduction, there are the drastic variations because the two different organisms are involved, right? The male or the male gamete comes from the male, and whereas the female gamete comes from the female. So I have told you that in sexual reproduction, greater diversity will be generated. Why greater diversity will be generated? Because here, as I've told you, the two different DNA from the two different organisms are involved. While forming the zygote, the DNA of the two different organisms crossover and the information goes to the offspring. So depending upon the nature of the variation, different individuals will have different advantages. Now, there is the importance of variation. Variation is the key factor for survival. They form the basis of the hereditary. Adaptability to the adverse condition is brought about the variation. That's why, as I've told you, that variation is the key factor which will help us in surviving the changing environment. Some variations are very useful. They help an organism to adjust. But there are some variations which somehow are not useful. New varieties of an organism arises due to the genetic variations. Right? Now, we have discussed about that. So these variations which occur over the period of these variation does not occur in a single day. They occur over the period of the time, and the variation in case of asexual is very less, whereas the variation in case of sexual, there are more variations in case of sexual reproduction. Okay, now we have to discuss about that. How do these variation pass on from one generation to another generation? What are the rules for inheritance of these variation or inheritance of the hereditary transfer? What are the rules? How characteristics are transmitted from one generation to another generation. Let me tell you that traits or characteristics which are passed on from parents to their offsprings or from one generation to another generation are controlled by gene. All the information which passes on from one generation to another generation, that passes through the genes. And where are the gene located? They are present in the chromosome. As I've told you that if we describe about the structure of cell, if I discuss about the structure of cell and what all, just a moment. If I'm describing about the structure of cell, let this be the cell, and within the center of the cell lies the nucleus. Within the nucleus, when the cell is still not ready to divide, there is the entangled thread-like structure. This is called as chromatin material. When the cell is ready to divide, at that time, I have told you yesterday also that it tend to form the proper thread-like structure. These thread-like structures are known as chromosomes, and the gene is present in these chromosome, right? It is so the inheritance, the main at functional unit of hereditary, which is present in the chromosome of the cell nucleus. It's this gene is made up of a molecule which is called as DNA. The gene is made up of a molecule which is called as DNA. As I've told you that when this chromatin material, when it detangles, it tries to form the thread-like structure, proper thread-like, which is known as chromosome. And chromosomes contain the small units which is known as gene. And what is gene made up of? It's the functional unit of hereditary which is present on the chromosome. It's composed of DNA. What is DNA? DNA is a molecule, and that is deoxyribonucleic acid, right? So DNA encodes from, it determines the particular, so it is this gene which is the unit of hereditary because it is the one which transmits the characteristics from parents to their offspring, right? Before we discuss about the rules for inheritance, we have to understand the various terminologies which are used. Now, the first terminology, that's what I say, that there is this gene. Gene is the unit of hereditary. Gene is the unit of hereditary which is present in the chromosome, and chromosomes are present in the nucleus, right? So next, the next term which we are going to discuss about is there are two types of gene. uh, Gene can be named by one more name, that's called as allele. Gene is also called as allele. Allele. One of the different forms of the particular gene. Okay, there is dominating gene and recessive gene. Now, what is dominating gene and what is recessive gene? We have to understand that first. Let me tell you the gene which will decide the characteristic of an organism in presence of a identical gene, that is called as dominating gene. Let Okay, let me tell you that gene always work in pair. Gene always work in pair to decide the characteristic of an organism. For example, if we talk about uh, hair color of eyes, that will not be decided by on that will be decided by one gene, but there should be a pair of gene for each characteristic. For example, if there is a color black, then there should be pair of gene for each characteristic. Gene always work in pair. You have to keep this thing in consideration, that gene will always work in pair. Now, out of these, there can be a different type of gene. One is dominating gene and one is recessive gene. But it is not always that the dominant and recessive both will be present in the same setup. That we will discuss. But what is a dominating gene? Dominating gene is that gene which will decide the characteristic in presence of a non-identical gene. Means in the pair, if there is non-identical gene which is not similar, if there are B and B, for example, they are same to same. If there are, right, they will be called as identical genes. These two pairs, if I'm talking about this pair and this pair, they will be identical gene because both the genes are same. But here, this is a non-identical pair. This is a non-identical pair because these are two different gene governing only one characteristic, that is color of hair. Two different gene governing one characteristic, that is color of hair. Right? So, that gene which decide the characteristic in presence of a non-identical gene, that we call as dominating gene. Okay, and the gene which decide the characteristic only in presence of an identical gene, that's what we call as recessive gene. All right, now we are going to discuss about one more term, that's called as genotype. The written description of the genes. The genes are written by using the alphabets. Let me tell you, when we present the written description, that's called as genotype. But for written description, we are using two type of letters. One is capital letter and one is small letters or alphabets. Capital letters indicate the dominating gene, whereas the small letter will indicate the recessive gene. So while we give the written description of the gene, that's what we call as genotype. The genetic composition of an individual is called as genotype, whereas the characteristic which they express, that is what we call as phenotype. So there are gene always work in pair. Where are the genes present? Genes are present in the chromosomes. Where are the chromosomes? Chromosomes are the one when the chromatin material which is present in the nucleus gets detangled. At that time, they form a proper thread-like structure which is known as chromosome. Genes are the units of heredity. Why? Because they pass on the information from one generation to the another generation. Genes always work in pair, right? So there are two type of gene, dominating and recessive. In the pair, if there are two different gene which are not same, but they are governing the same characteristic. For example, they are governing the height. As you know that height of an organism can be either you can say dwarf, the small, okay, or it can be tall. Both these characteristics govern this, that's height, right? But the characteristic which will be prevalent among the maximum population, that is what we call as the dominating. The characteristic which is present in the maximum population, that's dominating gene. That's the dominating characteristic, and dominating gene is always written with the capital letter. Even the dominating gene can also be said as that whose phenotype will be expressed even in presence of another allele, or even in presence of a non-identical allele, non-identical gene, which decide the characteristic in presence of a non-identical gene. That's what we call as dominating gene. Right? So I hope that I am quite clear on these terms. Before we discuss about that, what all was given by Mendel. Right? So as told you that the inheritance of the characters is due to the fact that both the father and the mother contributes equal amount of genetic material. So for each traits there are two factors. Two factors. Factor name was given by G.J. Mendel, whereas later on this factor was known as what we today know as gene. One gene is from father and one gene is from mother. Okay, so G.J. Mendel conducted the experiments to study about this. That's why G.J. Mendel is known as father of genetics. Why he is known as father of genetics? Because it was the Mendel who worked out the main rules for inheritance. The rules for inheritance of the traits in human beings are related to the fact that both father and mother contribute an equal amount of genetic material to the child. This means that each trait can be influenced by both parental DNA as well as maternal DNA. So it was that's why he is known as father of genetics, because he was the first person to study about the basic laws of inheritance. But before I discuss about the basic laws of inheritance, we have to understand the certain terms and terminologies which we have discussed. You can see that on your screen. These are the thread-like structures which is known as chromosomes. But chromosomes are not visible every time if we see the cell. You will see only the genetic material. That is, if the cell is in the non-dividing stage, we can see only the genetic material. That is the entangled thread-like structure, that's called as chromatin. But once the cell is ready to divide, so that it will form, at that time, The entangled structure detangles itself, form the proper thread-like structure which is known as chromosome. Then chromosome, as I've told you, as the units of hereditary. Chromosome contain the units of hereditary which is known as gene, and gene is composed of a molecule which is called as DNA. On your screen you can see the structure of DNA. It's a double helix structure composed of the various nucleotide fibers, right? Okay. Next, so to know about that, how the inheritance take place and why the inheritance take place, it was done. The experimentation was conducted by G.J. Mendel. Now, Mendel has selected a particular plant, and there was a reason why he selected the particular plant to study about the characteristic. If he would have studied the humans, it was not possible for him to study the different generation of the human. So for that, he studied, Mendel opted for garden pea, or which is known as Pisum sativum, to conduct his experiments, and there was a proper reason for that. And the reason was that the pea plants or Pisum sativum grow quickly and it is easy to study. Why? Because there are distinctive characteristics which can be visible to a layman also. Pea plant shows such characteristics which can be even identify the layman. You don't require a scientific knowledge to study about it. There are the characteristics present in this pea plant which can be identified by anybody. So that's why he studied, he used pea plant. Why? Because first of all, it grow more quickly. Second, it's easy to study the characteristics of the pea plant. Third reason is that pea plants show both the type of pollination. It can self-pollinate and has flower structure. And even if we want, it can cross-pollinate also, right? And has the proper flower structure so that it was easy to study. Third reason is that garden pea has number of easily detectable contrasting characteristics, which I have told you that that's the main reason why he selected pea plant. And he kept record of each and every observation which he has seen. Now, what were the main characteristic on which G.J. Mendel worked? And the main characteristics which are easily identifiable by the even layman among the Pisum sativum is that size or height. Height was one of the factors. Height was one of the characteristics which was studied by G.J. Mendel. Second was position of flower on the stem, right? Third was shape of the pod. What we mean for pod is that the जो मटर की फली होती है, that's what we call as shape of the pod. Then shape of the seed, because there are some seeds which are quite round, but there are some seeds which are wrinkled one, which are wrinkled. So this characteristic is also visible. Once you take the seed of pea in your hand, you will be easily able to identify there are some seeds which are round and there are some seeds which are wrinkled. So this also can be easily identified. This characteristics can all easily identified. Now, in case of if it would have taken some wheat plant or the rice plant or some other plant or mango, you can see that there are not too much distinctive characteristics which can be easily studied. Why? Because it's the size of the rice is almost same, the size of the wheat grain is almost same, color is same. Here we can see that there is difference in the height of the plant, there is difference in the flower where it is present. Is it present on the terminal, at the terminal, or is it present on the axle? Then color of the pod, shape of the pod, that is it is inflated or constricted. Constricted means whether it is like this, it is quite like this, right? So, or whether it is inflated. Color of the seed, there are color of the seed which you can easily identify because the seeds of the pea plant are available in two colors, one is yellow and one is green. So that is also the characteristic which can be easily identified. And color of flowers, color of flowers are either violet or white. So that is also which is visible, which is easily visible to even a layman can also understood that all these characteristics, all the differences, all the variations. So that's why G.J. Mendel used pea plant for studying the rules of inheritance.